Okay, let's start today's lecture. So before the start of today's lectures, let me brief about the previous lecture. So in previous lectures, we have basically discussed about the parathyroid glands, their secretion, then the transport mechanism of parathyroid hormones, and the mechanism of parathyroid hormone release. So in today's lectures, I will be basically focusing mainly three things. One is the calcium homeostasis mechanism, and how these parathyroid hormones are involved with the regulations of mainly calcium and phosphorus, and after that I will discuss about what is the role of calcitonin in calcium homeostasis, and after that we will discuss about the abnormalities of parathyroid gland. So let us start with the first part, that is about the calcium homeostasis. Yes, it is very important for in physiological point of view that body needs an optimum amount of calcium to perform various type of physiological function. So to maintain this optimum amount of calcium, parathyroid hormones played an important role. Not only parathyroid hormones, we could say there is another important molecules that is also important, that helps to maintain the balance of calcium, that is the vitamin D. Now, how this calcium homeostasis actually takes place? See, the calcium homeostasis actually depends on the amount of calcium available into the circulating blood, as well as the available amount of parathyroid hormone. You can see when blood calcium is low, calcium sensing receptors in the parathyroid glands are inactivated, leads to release of parathyroid hormones. So the first condition is in the circulating blood the calcium level is not normal, it is less than normal amount. So in that particular conditions there is an activation of a particular receptor that is called CASR or calcium sensing receptors. So in the homeostasis mechanisms, the activations or inactivations of calcium sensing receptors is very important, and these calcium sensing receptors basically respond based on the amount of calcium available in the blood plasma or in the circulations. Here, after the release of parathyroid hormones, it stimulates mainly three things. So what are the most important things that is carried out by parathyroid hormones? The first one is the resorption of calcium that takes place into the bone, right? The second thing is calcium reabsorption, and this calcium reabsorption takes place into the renal system, right? Not only the resorptions and reabsorptions are the most important things happens during calcium homeostasis. Another important thing that is this parathyroid hormones upregulates the activity of a particular enzyme, that is 1-alpha hydroxylase enzymes. And this 1-alpha hydroxylase enzymes is very important because it involves with the activations of an inactive vitamin D to its most active form, right? So that we could say this parathyroid hormones is very important because it involves with the resorption process, it involves with the reabsorptions, as well as it involves with the activations of a particular enzyme that facilitates the formation of active vitamin D from inactive one. You can see this vitamin D then acts on the bone and kidney to increase the calcium resorptions. So here we could see the resorptions that occurs into the bone, which is facilitated by this vitamin D, and reabsorptions that is also facilitated by this. It occurs into the renal systems, which I am going to discuss into the diagrams. Along with this, if you see the other important features, functional features of parathyroid hormones is it involves with the regulations of calciums into the intestines, right? Generally, we know the most important source of vitamin D is sunlight. So sometimes we can extract the vitamin D from dietary foods. So the extractions of the vitamin D from dietary foods is also influenced by this parathyroid hormones. Now you can see from here, this is the diagrammatic representations of calcium homeostasis. You see at the initial point of time or initial parts of these presentations, the levels of ionized calcium is very less or low. These conditions lead to the secretions of parathyroid hormones, and these parathyroid hormones takes part several physiological actions, as I have already mentioned. One is resorption, another one is reabsorption, and another one is the activation of a particular enzyme which helps in the conversions of inactive vitamin D to active vitamin D. You can see the parathyroid hormones involves the calcium resorptions from bone. These parathyroid hormones also facilitates the calcium reabsorptions from the urinary filtrate, right? Other than this, the same hormones act into the enzyme which helps in the conversions of this inactive form of vitamin D to active form, right? Once it gets activated, it facilitates both the resorptions of calcium from bone and reabsorptions of calcium from this urinary filtrates. Along with this, as I mentioned, vitamin D or active form of vitamin D now comes to the GI tract, and here it not only acts into this, here in this particular case, gut helps to absorptions of calciums, as well as from what is the most important sources of the calcium absorptions in the gut, it is the dietary calciums, and this absorptions is facilitated by this vitamin D, right? It means ultimately these are the most important pathway by which it helps to maintain the calcium level in the blood plasma. Initially it was very less. Now if you see the calcium level into the circulatory blood, it is more than initial point. Now we have another important mechanisms called negative feedback mechanisms. These negative feedback mechanisms helps to maintain the balance of or optimum amount of calcium into the flowing blood. You can see here the ionized calcium levels is very high, and these conditions inhibits the activations of the formations of active form of vitamin D, as well as it also prevents the release of PTH from parathyroid glands. So this is an overview by which the calcium homeostasis can take place. Now, as I mentioned that the parathyroid hormones mainly involved with the three tasks. Now question is, what is the mechanism of the resorption? What is the mechanism of reabsorption? That I am going to discuss in the next slide. See here, the mechanism parts I have already started in the previous class. Let me continue the same parts first. Here you can see the most important part that is highlighted here, the role of parathyroid hormones, their target organs and physiological effects. So the role is the calcium renal reabsorptions, calcium mobilizations from bone, as well as intestinal absorptions, that is called the indirect mechanisms. Along with this, another important functions of these hormones in the target organs is to increase the activity of the enzymes, as well as to change or alters the renal reabsorptions of phosphate. Here there is an increased excretions of phosphate takes place into the renal systems in presence of parathyroid hormones. I am going to discuss about that. So for all these cases, the most important thing is there must be specific receptors into the target cell so that parathyroid hormones can act into the target cell. Right? The question is, what are the different type of receptors that are present into the target cell or that are present into the target cell membrane? Yes, according to the latest research, there are multiple numbers of parathyroid hormone receptors that are found in target cells or target organs. In some cases, the functional features of all those receptors are explained properly, and in some cases it was found that the functional features of some receptors are still unknown. So if you see the most important receptors that are located into the target cell membranes, these are parathyroid hormone receptor 1, 2 and parathyroid hormone receptor 3. So out of these 3 receptors, the most important one whose functions is known is parathyroid hormone receptor 1, right? Regarding the functions of 2 and 3, there are lots of controversial issue. What is the exact functional role of these receptors is still controversial. So we are going to discuss about the role of parathyroid hormone receptor 1, or how this parathyroid hormone receptors 1 takes part into the resorption process, reabsorption process. This is already discussed in previous lectures, that the first part is parathyroid hormone effects on renal calcium reabsorption. So before discussing about, we must know about the locations of those receptors that takes part into the reabsorption process, right? You can see this is the luminal surface or apical surface of distal convoluted tubules of nephron, and this is the basolateral parts of the distal convoluted tubule, right? Here, when the structures are studied, mainly it was found that there are mainly 2 channels that takes place into the reabsorption process. The first one is present in the luminal surface and another one is present in the basolateral surface or basolateral membrane. So what is the features of these channels? The one channel is involved with the transport of calcium ion and another one acts as an exchanges. So what exchanges? It is calcium sodium or sodium calcium exchanger. Now the activity of both the channels and exchanger depends on the availability of calcium into the circulating bloods, depends on the availability of parathyroid hormones, right? So as I mentioned, when the level of calcium ion is less into the flowing bloods, so there will be hypersecretions of parathyroid hormones, and that parathyroid hormones helps in the reabsorption process from reabsorptions of calcium from renal filtrate. So you can see here at the initial point of times there is an influx of calcium ion in presence of parathyroid hormones, right? So why there is an influx of calcium? As because there is a less amount of calcium into the circulating bloods, which stimulates the parathyroid glands to release parathyroid hormones. So here there is a more amounts of parathyroid hormones that stimulates the influx of calcium ion, or so called that stimulates the reabsorption process, right? Not only this parathyroid hormone is sufficient in the influx of calcium or reabsorptions of calcium. This is also influenced by some other factors that I have already mentioned. What is the factors? That is the vitamin D, right? So how this vitamin D is helpful in the reabsorption process? The vitamin D actually helps to produce a special type of proteins, or that is called calvinin D proteins. And not only this, it facilitates the complex formations between these proteins and calcium, right? You can see there is a formations of complex between this protein and calcium, which is influenced by vitamin D. And after the complex formations, this is also discussed that how this complex comes towards the basolateral membranes. So as the concentrations of this complex is more central positions, it moves towards the basolateral membranes by the process of diffusions, right? And it comes to the basolateral membrane ultimately by the exchanges it is diffuses, reabsorbed into the interstitiums, right? Now here the most important things, the exchange of calcium and sodium needs some energy molecules, and these energies are provided by the calcium ATPase, right, which provides the energy that allows the reabsorptions of calcium into the interstitium. This was the basic mechanisms how the renal reabsorptions of calcium takes place, and what is the role of parathyroid hormone and role of vitamin D here. I hope this part is now clear. Another important part, as I mentioned, that the parathyroid hormones not only involved with the reabsorptions of calcium, it also helps to maintain the optimum amount of phosphate, right? Or it helps to maintain the phosphate amounts in flowing blood. So here you can see parathyroid hormone effects on renal inorganic phosphate reabsorptions. Here it is little bit different, the mechanisms is little bit different from previous one. So how it is different? In this particular case, the most important things involved is some transporter. So what is the most important type of transporter that are involved with the reabsorptions of inorganic phosphate? These are sodium phosphate co-transporter, right? So what is the most important things here? The most important thing is the transporters, right? If you see the natures of the transporters, there are multiple number of transporters are present into the proximal tubules of nephron, right? But not all the transporters are involved with the regulations of phosphate reabsorptions, right? The question is, what are the different types of transporter that are present into the proximal part of renal tubules, and which one is the most important one that involved with the phosphate reabsorptions? See, PTH, that is parathyroid hormone, decreases the renal reabsorptions of phosphate by decreasing the expressions of type 2 sodium phosphate co-transporter. Yes, in previous case what we have seen, parathyroid hormones increase the reabsorptions of calcium, but here parathyroid hormone decrease the reabsorption of phosphate. Both the reabsorptions of calcium and phosphate occurs into the tubule. One occurs into the distal part of the nephron or convoluted tubules, and one is the proximal part. It means there is a structural difference as well as some functional difference also occurs into this reabsorption process. You see, in kidney, PTH acutely decrease the expressions of sodium phosphate co-transporter by stimulating their internalizations via coated vesicles. Yes, see here, this is the proximal tubule, right? In this proximal tubules, if you see the type of co-transporters, mainly 3 different types of co-transporters are present into the proximal tubule, right? One is sodium phosphate co-transporter type 1, then type 2 and the type 3. So according to research, it was found that the most important type of co-transporter that involves with the reabsorptions of phosphate is the transporter 2, that is the type 2A, this one, right? So what is the exact mechanisms? So you can see here, as I mentioned, the level of PTH. PTH decreases the renal reabsorptions of phosphate. In the circulating blood, PTH is available. So that PTH reduces the reabsorptions. How? There is the internalizations of this type 2A co-transporter, right? This is type 2A co-transporters. There is the internalizations of type 2A co-transporters, and after the internalizations, it is attacked by lysosomal enzymes, right? These lysosomal enzymes attack leads to the degradations of this type 2 co-transporter, right? These attack of type 2 co-transporter by lysosomal enzymes leads to the degradations of this co-transporter, means there is a decrease of expressions of this type 2 co-transporters. As I have mentioned in the first slide, PTH decreases the renal reabsorption of phosphate by decreasing the expression of type 2 sodium phosphate co-transporters. So how there is a decrease of expressions? The decrease of expressions depends on the internalizations of the transporters and the lysosomal breakdown of that transporters, right? See here, PTH bindings to its receptor initiates the signaling pathway leading to the membrane retrieval followed by lysosomal degradations of these transporters. The decreased expressions of the transporter results in decreased phosphate reabsorptions. Obviously, if there is a decreased reabsorptions of phosphate, it means there is a hyper excretion of that phosphate through urine. That is mentioned here. Decreased phosphate reabsorption means increased excretion of phosphate through urine. Both the process are carried out by the level of excretions of that co-transporters. If the level of excretions is less or decreased, then it will reduce the reabsorption of phosphate. Thus, unlike other transporter such as aquaporin 2, once the sodium phosphate co-transporter is internalized, it is degraded, and this is an irreversible internalization. Means once it is degraded, there is no chances of involvement of that co-transporter in the reabsorption process. Clear? So that is why it is mentioned. As I mentioned, this is an irreversible process. Means once the transporter are degraded, it will not take part into the reabsorption process further, right? So ultimately, it reduces the reabsorption of phosphate. Another important factors that also takes part into the reabsorption process. What is this? Phosphate reabsorptions in the proximal tubules can also be decreased by fibroblast growth factor 23, that is FGF23, right? So what is the role of this FGF23 and what is the actual exact chemical features of this FGF2? It is nothing, it is basically a peptide, right? And this peptide are produced from a particular cell types. So what is the most important cell types that helps to synthesis of this FGF2? Mainly the synthesis occurs into the bone, mainly the osteoblast as well as the osteocytes. Both the osteoblast and osteocytes takes part in the synthesis of this FGF23, and this FGF23 also involves with the reductions of phosphate reabsorption. So how does FGF23 involved with this reabsorption process? Yes, FGF23 suppresses 1-alpha hydroxylase activity. As I've already mentioned that 1-alpha hydroxylase enzymes is an important enzyme that takes part into the conversions of inactive vitamin D to active vitamin D. And this active vitamin D also takes part in the reabsorption process. But here, there is an inactivation of these enzymes by fibroblast growth factor 23. So due to the inactivations of these enzymes, there is no chances of conversions of inactive form of vitamin D to its active form, right? So ultimately, it affects the amount of available vitamin D. At the same time, it also affects the expressions of co-transporter, that is sodium phosphate co-transporter. It reduces the expression level of sodium phosphate co-transporters. So now let me show you the diagrammatic representations. See here, as I mentioned, what is the most important sources of that FGF23? See, the FGF23, the most important sources is the osteoblast and osteocyte, and once it is released from this bone or osteoblast, right, it inhibits the action of 1-alpha hydroxylase. See, this is the FGF23 which is released from bone, and after release it shows two mode of actions. So what I am discussing here about the inhibitory actions of FGF23. It inhibits the action of 1-alpha hydroxylase that ultimately leads to the effect, or it affects the conversion of 25 hydroxy vitamin D, that is the provitamin D, to active form of vitamin D, right? Not only this, once it inhibits the actions of these enzymes, it promotes the excretion of phosphate called phosphaturia. It means the level of phosphate into the urine is very high. Means the excretion of phosphate in urine is very high, instead the reabsorption. Clear? This is how fibroblast growth factors is involved with the regulation of phosphate into the blood, right? Another important part that I mentioned is about the transporter excretions. See, here there's a more excretions, but with the times the level of excretions is decreased due to the lysosomal attack, right? So that also affects the reabsorption of phosphate molecules. Other than this, there is other transporters also present. But if you see their functional features, see, type 3 co-transporters are most likely located at the basolateral membrane and play general housekeeping role in ensuring basolateral inorganic phosphate influx if apical entry is insufficient to satisfy cellular requirements. Yes, generally what we see, the type 2 is mainly involved with the regulation of optimum phosphate level. But what they've discussed here, suppose somehow there is a disturbance of influx of or disturbance of phosphate reabsorptions through this type 2. So what is the alternative way? Yes, there is another transporter which played an important role as a housekeeping role that helps to maintain the balance of phosphate. That is why it is mentioned. It ensures the basolateral PI influx if apical PI entry is insufficient to satisfy the cellular demand. Clear? So this is what is the role of this type 3 co-transporter. Clear? Another important point, basolateral exit, which is necessary to complete transcellular PI reabsorptions, is not well defined. As I mentioned, there are a number of co-transporters. Till date, the functional features of some transporter are well established, and there are some controversial issues too. So that is why it is mentioned the basolateral exit, which is necessary to complete transcellular PI reabsorptions, is still unknown or not well defined. So this is what is the exact mechanisms of phosphate reabsorptions, or how the balance of phosphate in the flowing blood is maintained using, by changing the expression pattern of different type of co-transporters and involvement of different type of peptide like FGF23. Now, the reabsorptions of calcium part are done. Then reabsorptions of phosphate part is also done. Now see, what is the other important role of parathyroid hormones? See here, as I mentioned, parathyroid hormones also involved with the resorption process, right? And resorption basically occurs into the bone, right? It means the parathyroid hormones has direct actions into the bone. See here, in bone, parathyroid hormone releases calcium from stores that is readily available, and at the same time, after the release of parathyroid hormone, calcium hormones from readily available bone, subsequently PTH stimulates the release of calcium further. So by some other mechanisms, right? And this release of calcium is nothing. This release of calcium from bone is called the resorption process or resorptions of calcium. Now, during the resorptions of calcium, some chemical event takes place. These chemical events ultimately alters the bone structures called bone remodeling or remodeling of bone. So mainly two events occurs into the bone remodeling process. The first one is, you can say, bone remodeling involves bone formations by osteoblast and bone resorptions by osteoclast, right? It means the formations of osteoblast and resorption of osteoclast are the steps of bone remodelings, and here how this bone remodeling is facilitated by PTH. See, parathyroid hormone stimulates both the aspect of the remodeling process, right? And bone remodeling ensures bone repair and is necessary to maintain calcium homeostasis. Yes, what we see in the resorption process in bone leads to the release of calcium from bones to the circulating blood or flowing blood. And at the same time, we know the calcium is very important for the formations of bone. Clear? If there is a hyper release of calcium or hyper resorptions, obviously the porosity of the bone will be decreased. Increased. Osteoporosis may happen. So now we have a specific mechanism that helps to maintain the balance. This is called remodeling process. So what is the exact process? See, PTH stimulates the osteoblast first. So once it stimulates the osteoblast, it secretes some special type of molecule. One is macrophage colony stimulating factors, and another one is receptor activated, that is nuclear factor kappa beta, that is a ligand protein. So both mainly this ligand is important into the remodeling process. So how this ligand protein is important? This stimulates the pre-osteoclast to differentiate into osteoclast. This is the pre-osteoclast or precursor osteoclast, right? So the formations of this precursor osteoclast is facilitated by these ligands, and these ligands expressions or formations of these ligands are stimulated by the available PTH, right? See, whenever the precursor osteoclast levels is increased, it undergoes a differentiation process. Ultimately, it produces the osteoclast, and as I already mentioned, the resorption takes place from the osteoclast cells. So it means there is the release of calcium from the bone matrix of osteoclast, right? At the same time, as I mentioned, this remodeling also involves the formations of the bone, formations of osteoblast, right? And these formations of osteoblast is facilitated by some growth factors, and that growth factors are also released from that osteoclast cells. See, these released growth factors initially produce the precursor of osteoblast, and it undergoes again through a maturation phase, and that promotes the formation of osteoblast, that is the cell of bone. Clear? So it means it is cyclic events. And how these cyclic events are regulated? The cyclic events are regulated by the influence of the available PTH. In one pathway, it releases calcium, and in another pathway, it helps to produce the osteoblast cells, which is an important cell types of the bone, right? In both the cases, the most important factors, one is the Rankel's ligand factors or proteins, and another is the growth factors. Both the factors are equally important to maintain the remodeling process of bone. Here you can see the role of vitamin D, active form of vitamin D. These active form of vitamin D are produced by the positive stimuli of enzyme, and once it forms, it acts into the osteoblast, and there is an expression of this ligand protein, and this ligand protein binds and has to produce the precursor osteoclast, that is the pre-osteoclast, and ultimately mature osteoclast, and mature osteoclast is the important source of calcium as well as the phosphate. So it means the vitamin D indirectly involved with the reabsorption or involved with the reabsorptions of both the resorptions of calcium and phosphate from bone. Now see, what about the type of mechanism that I have discussed now, that all are related with the parathyroid hormones, right? Now, there is an important hormone, that is the calcitonin hormones, which is not secreted from parathyroid. It is basically secreted from thyroid glands. Now, question is, what is the role of calcitonin here, or how this thyroid glands involved with the homeostatic balance of calcium or ionized calcium? See, when the amount of calcium in the blood rises above the normal range, the important cell type, that is the parafollicular cells in the thyroid gland, secretes calcitonin, right? So here the condition is the level of calcium. It is not the normal, it is more than normals. In that particular conditions, there is a secretions of calcitonin from parafollicular cell of thyroid. This calcitonin decreases the calcium levels. Yes, we have to maintain the balance. Not more than optimum, not less than optimum. Whenever it is less than optimum, the parathyroid hormone played an important role to maintain this. When it is more than optimum, the calcitonin hormone played an important role. See, what is the most important role of the calcium? Calcitonin. Calcitonin basically inhibits the formation of osteoclast, right? Generally, PTH and vitamin D promotes the osteoclast formations. Here, calcitonin inhibits the osteoclast formations. So, as there is inhibition of osteoclast formations, there is no chances of resorption, right? It will helps to protect the bone by maintaining the level of calcium, as well as you can see what are the other important features of calcitonin. It inhibits calcium reabsorptions in the kidney, and it promotes the depositions of calcium in the bones, as well as it inhibits the absorption of calcium into the intestines. It lowers the calcium, ultimately it lowers the blood calcium levels. So the role of calcitonin is just opposite of the role of parathyroid hormones. So this is how the homeostatic balance of calcium can be maintained. Now, under certain pathological conditions, what we have seen, either there is a hypersecretion of hormones or hyposecretion of hormone. Means both the hypo and hypersecretions are nothing. This is the disorder of the gland, right? So, what are the disorders of this gland, right? Yes, the disorders are basically classified based on the activity of the gland. So, this is hyperthyroidism, parathyroidisms and hypoparathyroidisms. So, what is the conditions that may develop during hyperparathyroidisms? See, this conditions is developed when an individual produces too much PTH hormone, right? So, if the PTH levels is high, how it alters the calcium? Overproduction of the hormones releases the large amount of calcium, right? And which causes hypercalcemia. So, this is the pathological conditions that is related with the disorders. And in this particular case, what we have seen, there is a large amount of blood calcium, right? Means the regulatory mechanisms is not working properly. That is why there is an imbalance of calcium into the flowing blood. So, what are the most important symptoms that is related with this hypercalcemia or hyperparathyroidisms? It includes extreme thirst, more urine productions, abdominal pains, and frequent mood change. Here, what is the negative impact of these conditions? See, if it is undetected, high level of calcium leads to bone thinning, that is the osteoporosis, and can also cause stones in the kidney. So, these are some related things of disorders of parathyroid glands. And at the same time, in hypothyroidism conditions, level of PTH is less, that leads to the development of these hypothyroidisms, and it is most important causes of hypocalcemia, and the most important symptoms are tingling sensations, muscle cramp, as well as spasms. So that can be again divided into primary, secondary, but the basic thing is this, that in some conditions there is a hyperactivity of parathyroid glands that alters the calcium level and that develops the disorder, that is hypo and hyperthyroidisms. So this is what that I have overallly explains from this particular competency. So let me brief again. In previous class, I have discussed about what are the most important cell types, what are the important hormone released, what is the transport process and the mechanism of hormone release. After that, I have discussed about the homeostatic mechanisms and what is the exact mechanism of reabsorption and resorption, and finally what is the disorders that is related with the parathyroid gland. Thank you.